Imagine discovering that the trillions of microscopic organisms living within your body could hold a key to predicting and battling one of the deadliest cancers. That's the startling insight emerging from a cutting-edge study on clear cell renal cell carcinoma, ccRCC, a prevalent urinary system malignancy that recurs in over 40% of patients post-surgery, posing a grave threat to survival. But here's where it gets controversial. What if our traditional focus on human genetics alone is missing a crucial piece of the puzzle? Stick around as we dive into how the microbiome might just redefine cancer prognosis and treatment, challenging long-held beliefs in the process. Clear cell renal cell carcinoma, often abbreviated as ccRCC, ranks as a major kidney cancer type, with patients facing daunting odds of recurrence after surgery. Standard treatments typically involve aggressive surgeries like radical or partial nephrectomy, paired with advanced drugs such as targeted therapies and immune checkpoint inhibitors, ICIs, to boost overall survival rates. Yet, up until now, our approaches to classifying and treating this disease have relied heavily on analyzing the human genome, the genetic blueprint within our cells. This overlooks a potential game-changer: the vast community of microbes inhabiting our bodies, known as the microbiome. Emerging research hints that imbalances in these microbial populations could fuel various diseases, including cancer. However, most studies have zeroed in on fecal samples from the gut, which might overlook vital details about microbes thriving in specific organs. And this is the part most people miss. Next-generation sequencing of tumor tissue has uncovered bacterial signals in various cancers through initiatives like the Cancer Genome Atlas, TCGA, hinting that the microbiome could emerge as a fresh diagnostic and prognostic tool. For beginners, think of it like this: just as a soil's microbial mix affects plant growth, your body's internal ecosystem of bacteria might influence tumor behavior in ways we're only beginning to grasp. Leading this exploration are researchers Xinhong Cui from Xinhua Hospital at Shanghai Jiaotong University School of Medicine, and Brian Rennie from Vanderbilt University Medical Center in the USA, along with their team. They delved into the ccRCC tumor microbiome's traits and its ties to patient outcomes and molecular traits by weaving together data from multiple sources, microbiome, metabolome, and transcriptome analyses across different centers. This integrative approach offers a novel lens for tailoring subtypes and therapies, potentially transforming how we handle this aggressive cancer. The investigators gathered tumor specimens and matched non-cancerous tissues, called paratumor tissues, from ccRCC patients who hadn't yet received treatment. They performed 16S rRNA sequencing, a technique that identifies bacterial species by analyzing a specific gene segment in their DNA, much like using a barcode to catalog different microbes, and untargeted metabolome profiling, which maps out the small molecules produced by cells and microbes. To ensure accuracy, they filtered out samples with low microbial presence that could be contaminated. The results? 16S rRNA analysis showed markedly higher alpha diversity in tumor tissues versus paratumor ones. For those new to this, alpha diversity measures the variety and evenness of species in a community. Here, metrics like feature count, number of unique microbial types, abundance-based coverage estimators, ACE, Chow-1 index, Simpson index, and Shannon index all pointed to richer microbial ecosystems in tumors. This suggests significant shifts in the microbial makeup within the tumor microenvironment, TME, the complex surroundings where cancer cells interact with healthy tissues and immune cells. Digging deeper, the team found fewer aerobic bacteria, those that require oxygen to survive, in tumor tissues, while anaerobic, oxygen-avoiding, and facultatively anaerobic, flexible with oxygen, bacteria stay about the same. This aligns with ccRCC's hypoxic nature, meaning low oxygen levels in the TME that foster such microbial adaptations. A volcano plot, a visual tool highlighting statistically significant differences, pinpointed bacterial genera, groups of related species, that varied between cancerous and normal tissues. To confirm bacteria's real presence, They use RNA fluorescence in situ hybridization, FISH, which tags specific microbial RNAs with glowing markers, combined with gram staining, a method to classify bacteria by their cell wall structure. These validations provided solid evidence of bacterial inhabitants in the tumors. Expanding their scope to a larger dataset of 428 ccRCC patients from the TCGA cohort, the researchers employ advanced clustering algorithms such as INT-NMF, consensus clustering, and similarity network fusion, SNF. These tools group patients into 3 distinct microbiome subtypes based on bacterial abundance levels. Survival analyses revealed stark differences in overall survival, OS, across these groups, with groups 1 and 3 facing poorer outcomes. Gene set enrichment analysis, GSEA, a method to detect overrepresented biological pathways, uncovered unique patterns. Group 1 showed ramped-up metabolism reprogramming, how cells alter their energy processes, and weaken immune activation compared to Group 2, while Group 3 had suppressed anti-tumor immune responses and atypical ccRCC metabolic traits. Adding whole exome sequencing, which scans for mutations across all protein-coding genes, revealed divergent mutation landscapes. Group 2 had the lowest rates of mutations in key genes like VHL, about 47%, and PBRM1, around 38%, whereas SCTD2 and BAP1 mutations topped 10% in all groups, linking these to issues like impaired metabolism and immune suppression. Metabolomic profiling further illuminated pathway differences, with glycerophospholipid metabolism, involving fats crucial for cell membranes, enriched in groups 1 and 2, and linoleic acid metabolism, related to essential fatty acids from diets, boosted in groups 1 and 3. Disruptions in linoleic acid pathways might signal worse prognosis, though ccRCC research on this is sparse, calling for more exploration. To give a relatable example, imagine linoleic acid as a vital nutrient for cell health. Imbalances here could be like throwing off the body's nutritional balance, potentially fueling cancer growth. In essence, this research carves ccRCC into 3 microbiome-driven subtypes, underscoring strong connections between microbes, tumor molecular features, and survival odds. It paints the microbiome as a potential standalone prognostic indicator, rivaling human genetic data, and paves the way for better risk assessment and customized treatments. Moreover, blending multi-omics data, microbiome, metabolome, and transcriptome, builds a sturdy platform to investigate how these elements interact. But here's where it gets controversial. By proposing the microbiome as a key player alongside genes, this study challenges the dominance of genomics in oncology. Critics might argue it's too soon to overhaul treatments based on microbes, given the complexity and potential for misinterpretation. Despite hurdles like a modest sample size and incomplete data alignment between metabolomics and sequencing, these discoveries set the stage for broader validations and deeper mechanistic research, potentially ushering in microbiome-informed advancements in neurological cancer care. For the full details, check out the original paper here. https://journal.hep.com.cn/fmd/en/1007/s11684-023-1029-3. What do you think? Could integrating microbiome data truly revolutionize how we approach cancers like ccRCC, or is this just another overhyped trend in medical research? Do you agree that microbes might be as crucial as our own DNA in fighting disease? We'd love to hear your take in the comments. Share your agreement, skepticism, or alternative views below.